Okay, so this is the third and final part of the Fragile X syndrome lecture series. Today we're going to be talking about potential pharmacological treatments of Fragile X syndrome based off of what we learned in the last lecture from the animal model studies. We're going to be talking about a few of the clinical trials that have attempted to target the mGluR system in human patients. But first, I just wanted to start off and just kind of think generally about genetic disorders such as Fragile X syndrome. And there are two main approaches that are being used in an attempt to treat Fragile X syndrome. You know, the first is since Fragile X syndrome is such a unique disease, there's only one gene that's involved. We know exactly what the gene is that causes it. So some people are thinking, well, maybe if we can just reactivate the affected gene, we could cure Fragile X syndrome. And currently, we aren't able to do that. It's quite difficult to do. But just thinking theoretically, if we could, you know, some people are attempting to be able to do this. And the second is reversing effects due to loss of FMRP. So people are trying to pharmacologically reverse some of the effects, and we'll focus on this in a little bit. But I first wanted to talk about some of the difficulties. So as I mentioned, currently we are not able to reactivate the affected gene, but even if we could, there are a few questions still at play. And the first is that, does development play a role? Is FMRP needed at some developmental time point? And if you reactivate the gene in the adult, FMRP may not, have that much of a beneficial cause if the critical period for development has already passed. Additionally, there might be compensatory mechanisms, in the adult. They didn't have FMRP their whole life, so the cells might have tried to compensate by changing some of the other signaling systems. And so if you reactivate FMRP, this um, might alter additionally some signaling systems. So uh, a few things to think about. There are of course, some difficulties when you're trying to reverse effects due to a loss of FMRP, either pharmacologically or however you do that. And the first is sort of the obvious one: can you truly reverse all of the FMRP-induced changes? For example, is mGluR signaling the only thing changed? So when we try and use drugs to alter the mGluR signaling, we're sort of leaving out the other systems that I briefly talked about in the other lecture. Um, and additionally, we're sort of relying on our animal models to accurately predict Fragile X syndrome when we try and target these specific systems. So if animal models have altered mGluR signaling, the human patients for some reason don't, then the pharmacological treatment will never work. Um, so nevertheless, as I mentioned, we can't currently reactivate the affected gene, so using pharmacological tools is our best option. And there have been um, two trials that I'll talk about that have attempted to target the mGluR signaling in human patients. The first is Phenobam. It was originally used as anxiolytic a long time ago, but then people realized it was a negative allosteric modulator of mGluR5. Um, and so they first tried it in preclinical studies in animal models, and they did show positive results. So this is a group that measured the filopodia and the density of these filopodia. Again, these are the long and thin, weak dendritic spines. In the knockout without treatment, you can see there's a higher density of these spines. Um, However, in knockout animals that were treated with MPEP, the inhibitor of mGluR5, or Phenobam, this restored the density of these filopodia to wild-type levels, um, and it also showed effects behaviorally. Uh, so they ran a clinical trial, a small clinical trial with six males and six females with Fragile X syndrome. There were no adverse effects, so the drug didn't harm the patients at all, and it did have some beneficial behavioral effects. The patients showed reduced hyperarousal and anxiety, as well as improved prepulse inhibition, which we talked about in the last lecture. Again, this is a very similar paradigm used in animals and human patients. So you can see the patients that were treated with Phenobam, so Phenobam minus baseline showed a higher pre-pulse inhibition compared to the patients that were tested and then retested under controlled conditions, without Pheneban. So it seemed to be beneficial. So a lot of groups got excited. This big drug company, Novartis, then took on these mGluR5 inhibitors and ran a much larger trial using this drug called mavoglurant. It was the first randomized, double-blind, placebo-based trial, so it was pretty well controlled. It was a much longer trial, so the Pheneban trial was actually just one administration of Pheneban. This was mavoglurant administered twice daily for 12 weeks long, and it had 175 adults aged, all with Fragile X syndrome, aged 18 to 45. They even separated some of the groups based off of their methylation status. So again, the gene is silenced by methylation, and in some patients they have more fuller methylation than others. So they were able to test this and even separate groups to look at. So they got the results back, and again, there were no adverse effects with these mGluR5 inhibitors. However, once again, or in this group, sorry, no beneficial cognitive effects were observed. So the big question was, why is this the case? So the first thing they thought of was, well, maybe these patients are too old for these mGluR5 inhibitors to be effective. So they were in a second trial a few years ago with 139 adolescents. So these younger patients aged 12 to 17 years. But once again, no beneficial cognitive effects were observed. So again, they're sort of forced to ask, why is this the case? They did run a relatively long trial, 12-week long trial, but perhaps longer drug treatments are required for this mGluR signaling to be reversed. Even though they used age 12 to 17 patients, maybe even these patients were past a critical period where mGluR5 inhibitors are effective, where FMRP is involved. So, maybe these subjects were still too old. But really, the drug company ended up dropping mavoglurant. They're not going to run any more trials. So it could be possible that mGluR inhibitors are not the best treatment option. The mGluR theory of Fragile X syndrome has been sort of the predominant theory for a long time now, but it hasn't seemed to translate well into human patients. And so I just wanted to leave off just again reminding everyone that mGluR signaling isn't the only thing that's involved here. There are other clinical trials going on for GABA receptor agonists, this other drug that is attempting to regulate dopaminergic signaling among others, as well as minocycline to attempt to treat this oxidative stress to neurons. And some of these other trials are showing some positive effects. But these mGluR5 inhibitors haven't seemed to work very well in the last few years. So I hope you guys learned something about mGluR signaling and Fragile X syndrome as a whole. If you guys have any questions, you can contact me in class. Thanks.